Gobind. I don't think we do that quite enough. So this has been absolutely fantastic. My first time in India, and it's just been a wonderful, wonderful experience. And in a way, I think Gobind has always been a very, very good ambassador for England, for India, because despite the fact he spent so much time abroad, I think he really kept his roots. And to us, I think he really showed, the richness of your culture and the warmth and humanity of your society. Just to tell you a little bit about my links with Gobind, I postdoced with him between 1985 and 1988, and that was a wonderful time because he'd switched to membrane proteins, and the gene synthesis was in full swing. There were gene synthesizers around, and what we could do is we could do cassette-based mutagenesis, which at that time was very new. At that time, you couldn't just order fragments of DNA, you had to make them yourself, and obviously this was a prime lab to do it. So when I arrived, really, we really started to do mutagenesis of proteins, and in particular of bacterodopsin. Scientific officer at Merck, we looked at folding of bacterodopsin because we could isolate and fold bacterodopsin. So these are really, really exciting times and really ambitious projects. And what it really taught me is, as a chemist, because I came to the lab as a chemist, is what you really need is in this new field, and it has been very exciting. I should say in that field, we're really lacking tools. So we don't have universal sequencing tools, we don't have polymerases, we don't have expression systems. So really, we really have to start from scratch. But as I said, what I'm hoping is that when we develop these tools, the biology of carbohydrates will just be as accessible as that is of nucleic acids. So really, we've spent the last sort of 10 years or so, developing analytical and synthetic tools in this area. And I want to talk in particular about analytical tools today. These carbohydrates are generally biologically relevant as glycoconjugates. So you know about biopharmaceuticals probably. So in red here you've got the sugars on EPO, human erythropoietin. And biopharmaceuticals, I think, is something Nico is going to talk about in a minute, so I don't want to dwell on it. You've got the cell surface carbohydrates linked to proteins or lipids, and we want to really understand the biology of those, and we need to know the structure of those and have probes for those. If you look at plant metabolites, a lot of them are glycosylated, and I'll talk a little bit about the analysis of that later on. And also we're very interested in sort of nanotechnologies, so making artificial glycoconjugates, be it DNA-encoded libraries, which is a big topic in pharma companies now, artificial lipid surfaces, and also solid surfaces, self-assembled monolayers, which are glycosylated. And what we're developing is generally enzymatic methods to make this, developing sort of enzymes akin to restriction enzymes to help us with the synthesis and the analysis. But as I said today, because I have very little time, I just want to talk a little bit about the analysis, this problem of carbohydrate sequencing. Any one of you who has worked with carbohydrates realize this is a real challenge still to analyze carbohydrates. So where are we with universal carbohydrate sequencing method? Wouldn't it be nice if you could sequence carbohydrates just as well as we could do with DNA? We think that would really push the area forward dramatically. So what's the problem? We obviously have fantastic sequencing tools, not only from the DNA field, but also from proteins. And mass spectrometry is particularly useful tool to deal with biological analysis, because you can't start with screwed samples, you need very little material, and we have got some beautiful, in particular, tandem mass spectrometry methods. And indeed, you can use them as well for carbohydrate analysis. So if you had, let's say, a disaccharide and you wanted to analyze that further, you could actually fragment that with collision-induced dissociation, and then do SMS studies and get some analytical data on the fragments, and then get some information on the sequence of this original carbohydrate. The problem is that mass spectrometry is completely blind to stereochemistry. So if you have isomers, for example, a glucoside and the galactosides, so these have completely different biological activity. all been streamlined, but for other glycans, this is really not possible at the moment. So really, this is a massive, massive challenge and bottleneck. And people always claim that carbohydrates are so complicated, and maybe this all doesn't mean anything. But to me, actually, carbohydrates just have a different store their biological information, their structure information is slightly in a different way. In comparison to DNA and proteins, carbohydrates tend to be actually much shorter. So you get a lot of biological information in much shorter sequences. And this biological information is much more dense. So the carbohydrate sequences encode these multi-dimensional biological informations, on stereochemistry, and because I come from the organic chemistry field, I find stereochemistry really elegant and fascinating. So I find that very attractive. As I said, because MS is the primary tool, we cannot distinguish between stereoisomers. So really, in a carbohydrate sequence, like say in a disaccharide, you not only have the information of sequence, so glucose and glucose, or glucose and galactose, and so on. You also have information due to the stereochemistry of these. These are all isobaric. They all have the same mass. But they have, as I said, very different biological information, and they only differ by this sort of one single stereocenter. Very, very elegant structural changes and changing biological information. Then you also store some information in the sequence. You've got alpha and beta stereochemistry, whereas the nucleic acid and the protein linkages are homogeneous. And also you can get different regioisomers. So really, with a dimer, you can have a lot of different biological information, and I think that makes this challenging, but also really, an interesting intellectual challenge. So, as I said, mass spectrometry is really the best method, but is limited. So the question is, can we, on the basis of mass spectrometry methods, add other methods that give us some information of the stereochemistry, and these methods we want to be quite universal. So what I want to quickly talk about is some method that we can add to mass spectrometry workflows, and that is called ion mobility spectrometry. And what ion mobility spectrometry is, is actually not a new method, I should say. We're just really borrowing this, from the mass spectrometry field for our purposes. But ion mobility spectrometry does is it measures three-dimensional shape by measuring something called collisional cross-section. So if you look at a single molecule, if it's very stretched out, it would have quite a large collisional cross-section. If it's very condensed, it would have a very small collisional cross-section, regardless of the fact that it might have a different mass. So the question is, can we combine this with mass spectrometry to get to this essential stereochemical information? Fortunately, there are now instruments off the shelf available. So, for example, this instrument here provided by Waters, and inside a mass spectrometer, there is already integrated this ion mobility tube, often used for other purposes, but actually this instrument could be harnessed for our purposes. And so what this tube does, it sends these ions, which we've generated in the mass spectrometer, so through CID, through this ion mobility tube, and depending on the collisional cross-section, you get different drift times, and you can separate them. So the question was, could we do this with carbohydrate? Could we fragment our carbohydrates and then get information on these fragments? So what we want to do is start with our different carbohydrates, which might have different mass. I'm using symbols here. So these represent just single monosaccharides. We do a mass selection to just select for one mass that might still be isomers, but they all will have the same M over Z. Then we do dissociation, and then we do mobility separation, and get information on the particular structure of these monosaccharides, put that into a database, and then reassemble the sequence through bioinformatics methods. And why do we want to do it at a fragment level? Well, one drawback for ion mobility is that it doesn't give you direct structure. It gives you this collisional cross-section. And generally, this is at the moment an empirical value, although you can actually calculate that using particular assumptions on configuration and so on. So what we still need is some standard. And the monomer standard we, for example, need for hexose is very small, but if we need standards for higher oligomers, then the numbers get very, very high. So really what we want to do is minimize the number of standards we need to assign the structure of these from the drift times. So this cuts a very long story short, and I don't want to show you a lot of data because I know this is not a mass spectrometer, but in a nutshell it works. So for example, here we have galactose and glucose. Galactose has this axial hydroxyl group, so it's a slightly smaller collisional cross-section, and the question was, could we see that with the instrumentation we have? And this just shows actually one of these spectrums we get from the ion mobility, and you can see very clearly that you can separate the glucose from the galactose ion. And we've done this with other hexoses, and in all the cases we're able to separate these. So we can truly, really distinguish between the different isomers here. So this is a very, very useful method to get at this stereochemistry of these particular building blocks. And this is, I think, for those of you who do LC, you might say, well, you don't get baseline separation. You know, this is not very good, but this is a very, very highly reproducible chromatogram. So if you use different samples, for example, we have two diglucosides. So these are dimers with two glucoside only actually differing in terms of alpha and beta. You can see that you get highly reproducible drift times in the ion mobility spectrum. So this is actually a very, very reproducible method, and the differences we see are statistically highly relevant. What we then noticed is actually, when we looked at the diglucosides, is that there seemed to be a memory effect on the linkage. We didn't really expect, because we cleaved the linkages here, and I should say we generate lots of different ion which increase the information we get from this type of analysis. When we looked at these different isomers, we expected actually to see identical drift times, because we thought we lose the information of the anomeric linkage. We thought this might be still a big challenge, a drawback, that actually the linkage information was lost. But what we noticed slightly unexpectedly is that the alpha anomer had this additional peak. So we wondered then, is that a sort of general feature that would allow us to distinguish with this method between alpha and beta anomers? We then measured systematically all the diglucosides, all the alpha-beta diglucosides, and we found indeed actually that is true. For all the alpha diglucosides, we got these additional peaks. So we get a different spectrum to the beta glucosides, so we can actually also distinguish between alpha and beta, which is very important when you do carbohydrate sequencing. I should say there was one outlier, which was this one here, which is a beta anomer, and we found actually that was due to an impurity. So we could actually pick up impurities of the other anomers through this method as well. So that now allows us, it's particularly useful for sequencing, getting de novo sequencing information, but it's also particularly useful because often we want to sequence glycoconjugates. So one of the problems we had was that we were generating through enzymatic methods peptides with different peptide sequence with either N-acetyl glucosamine or galactosamine. Again, these, for those of you who don't know sugar chemistry, these are really distinguished just by one stereocenter at the monomer form. These are totally isobaric structure. And the method here now allows us to start with a glycopeptide. And these glycopeptides also actually separate by ion mobility, but they're very difficult to compare to standards, and they give us quite complex patterns actually here, because we have different conformations. But if we do CID now, so to fragment this, we fragment off the sugar without needing any particular chemical fragmentation method, which is also a big advantage. of glycans, fragmented it by our fragmentation method, and then looked actually for the ion that represented this particular target structure, which we were really interested in. This is actually a terminal structure in many of these glycans. And we could see that when we treated it with our enzyme, actually we started with fetoin here, sorry, this is not on order, with fetoin, which has both linkages. When we treated it with, when we had a simple glycan, we could show that this here corresponds to one of the anomers. Our enzyme cleaved the other one off, and the third enzyme here cleaved mainly this structure off. So really, you can get sequence information, very selective sequence information from very, very complex samples. The last example I want to show is about plant glycosides. These are particularly difficult to sequence because we don't have any of these nice LC methods we have for N-glycans. And here we worked with some plant biologists, Birger Möller at Carlsberg Laboratories, who was interested in plant glycosides that had these alpha-hydroxynitrile A glycones. So these structures, so this is, hold on, this just tells me that I've done 20 minutes, so 5 more minutes and we finished. So basically, once you cleave the sugar off, this generates cyanide, and that acts as a defense mechanism in many plants, for example, in almonds. So they were very interested to get the structure of these glycosides. And some of them they had determined, and you can see they're very complex. And I think this is not uncommon. I'm not a plant biologist or biochemist, but I think this is not uncommon that you have plant metabolites. Notice there are also different glycones here. These nitriles come in different forms. But you also can see there's quite a lot of heterogeneity on the sugars. And what they really want to know is what is the sugar composition of these and what are the linkage types. And that's a very, very difficult answer to give. So what they normally do is they extract the plants, they do LCMS, then they do PrEP HPLC of the major peaks. The minor peaks they don't get enough material on, and then they have to do either NMR or they have to do chemical synthesis from authentic standards and compare them with LC methods. So this is a very, very time-consuming method. So the question was, could we use our fragmentation method now to really do de novo sequencing of these plant metabolites? And to finish off one paper, they had this one challenge that they had these glycosides of this natural product here, amygdalin, from almonds, and the LC gave them 3 peaks, and from the mass spectrometer, which from LCMS, they could show that there was a hexose linked to this structure here, but they didn't know which hexose, and they didn't know what the regiochemistry was, and they didn't know what the linkages were. But they realized that these 3 were actually isomers, 3 isomers of this general structure. So they challenged us to use the sequencing method to actually find that information. So we did the same thing. We started with our target structures. We could get them straight from their LC. And then we fragmented them in our mass spectrometer and did ion mobility separation. And what we obviously had already were standards that corresponded to substructures of this target structure. So we didn't have any authentic material of the target structure, but we had substructure. We had this amygdalin here, and we also had these diglucosides, which I've shown before here, which you potentially could correspond to these terminal structures. And by comparing fragmentation and ion mobility spectra of the target molecule with these standards, we could unambiguously show, and these are sort of the data which I don't want to go through. In fact, this was published in Biochemical Journal. We could actually tell them, that this here, they all 3 have glucoses attached to them, because it clearly showed that the ion mobility was corresponding to glucose. They were all beta linkages. Again, we can have this anomeric effect that we have the memory effect, and that this one here is linked to the 6th position, this is linked to the 4th position, and this is to the 2 position. And really, at the moment, we don't have any other analytical methods which could do this, at this sort of concentration from crude mixtures. So this is a really nice way of showing that, in principle, these methods are really giving us useful information. I should also say this also works with N-glycans. So we've taken this typical core pentasaccharide structure. With N-glycans, as I said before, we have other analytical methods, but it's a generic method that also works for N-glycans, and we can fragment that into these, and then we can compare that again to standards, structures, and all these collisional cross-section values correspond very, very nicely to those of standards, generated from completely different starting material, so it's obviously independent of starting material, and then you can assign actually the structure by building up from the fragments. Obviously that takes quite a lot of effort. So just to conclude our sort of vision for developing carbohydrate sequencing method, it's not that we maybe come up with a very, very simple method, but what we find is that incorporating ion mobility with already existing mass spectrometry methods means that sequencing, hopefully, will become much more generic. So we're at the moment, we get from mass spectra what we call sort of 2-dimensional information. We get already quite a lot of information on linkages, whether you have hexoses or hexnags, what the molecular weight of the building blocks is, whether it's branched or linear. We can do that already with existing mass spectrometry methods. But what we can't do is get the 3-dimensional structure, and at the moment we have to go through these protocols, and we need very, very large databases. There's currently a program funded by NIH which asks chemists to synthesize all the potential glycan structure in the human glycome. So these are 10,000 different structures. So we really need that if we pursue these sort of LC-driven processes. If we incorporate ion mobility, we propose that we go to the fragment level. We don't need the LC because ion mobility acts a little bit as an LC, but the beauty about ion mobility is we can do it on these fragment ions. We need much smaller databases, and hopefully that will really open up the area and allow us to develop much more generic sugar sequencing protocols, so that when people isolate carbohydrates, they can get very easy, very high resolution and accurate sequence information. I should say at the moment, you can see we're dealing with very simple structures, so I always compare the state of this sort of sequencing to the first commercially available TV, which is this one here, which was commercialized by the Baird company at Hastings in the UK in 1930. So this is what the first TV looked like. So I think we're still quite a way to match actually sequencing of DNA and proteins, but hopefully we're getting there by using this methodology. So I just have to thank really my team, in particular for the sequencing programs, Chris Gray, who was an absolutely outstanding PhD student, my collaborators who are mass spectrometrists, Claire Ayers and Perdita Baron, in particular Birger Moller for that collaboration on the plant project, funders, and I thank you for your attention.
Thank you, Sabine, for a beautiful talk, inducing us to do these simplified methods in the future.
Thank you.
Any questions? Please.
Very nice talk and very informative information related to the stereochemistry, because from the long time that carbohydrate chemistry are trying to solve this the problem related to the stereochemistry, variation of the stereochemistry of this glycan. Just two things I just want to know, that is it this technique, this ion mobility technique is still will be applicable for the biopolymers like xylan or starch, so these are the bigger the biopolymers, or it is only for the oligomers? And the second is, if it is for oligomer, then what the extent of the DP, the degree of polymerization that to which we can analyze the oligomers. So that is the thing. to think?
Yeah, that's a very good question. I mean, I think the polysaccharides will be a big target. I think we probably have to do pre-fragmentation of these, maybe through chemical methods, and then fragment at the fragment level. I mean, I think the DP, given that we deal with mass spectrometry here, I haven't shown a lot of other. But you can actually do the ion mobility on much larger fragments, as long as you have the standards. So you're absolutely right. The big challenge is in the polysaccharide field, and that's where we're moving. And this is why I showed this primitive TV. I'm not trying to sell this as at the moment state, ready to go. But polymer, we're looking. So we're working at the moment on polysaccharides. I think that's the really, really good challenge area. I agree with you.
Because either they have to be some chemically defragmented or something prior to probably to this kind of analysis.
Yeah, or enzymatic. It depends how big they are. Yeah, we can already do some fragmentations, but we haven't really done very much in that area. So that's sort of the next step. We focused on smaller structures at the moment, but it's sort of bottom up. But it's definitely something we're thinking about. Yeah, I think it's a very important application. Yeah.
Okay.
Let's give another hand to Sabine Flitsch. Thank you, Sabine. The next speaker is Nico Kalleveld, and he also was introduced as former chair already, and he will partly continue the topic that was discussed currently.
Thank you so much. It's a real honor to speak here for this fine audience, and I have to thank really the organizers locally for doing a splendid job. I'm representing a few people who do not travel much anymore, and so at Ghent University, where I work at the moment, I'm the third generation of people who have had some contact and training by Gobind. And so the other two people would be Walter Fiers, who was with Gobind in 1962 already. And he was the PhD supervisor of Roland Contreras, who was with Gobind in 75, 76. And Roland was my PhD supervisor. And I was sent to MIT for a brief while in 2000 or 99 it was, to try and help out with a particular challenge that the lab was facing at that time. So Walter really, to just illustrate the impact that Gobind has had also in our country. Belgium is a very small country, and the northern part is Dutch speaking. It's called Flanders, and we have a biotech institute there since about 20 years. And Walter was one of the founding fathers, really, of that institute. And there's now two centers, the Inflammation Research Center and the Medical Biotechnology Center, which I head at the moment, that really come directly from research lines that Walter has started up. And the Inflammation Center is built on the early work on cytokine gene sequencing that Walter did. And then the Medical Biotech Center is very much protein focused and derives from the sequencing work and the heterologous protein expression research line that Walter started very early on. So we're very, very grateful for that legacy in our country, really. So what I want to bring scientifically to you today is two stories that deal with, I think, big problems that act beyond the ribosomal stage of protein synthesis in eukaryotes. And so that would be processes that occur in the eukaryotic secretory system. And very broadly, conceptually, what happens in the eukaryotic secretory system is that after a protein has been translocated into the ER lumen through the translocon upon ribosomal translation, then glycans are attached, depicted here with these monosaccharide symbology that you just saw in Sabine's talk as well. So oligosaccharides are attached, N-glycans, to the asparagine side chains of particular sequence in the protein. And these glycans play essential roles. They're universally conserved in the eukaryotic world. They play essential roles in catalyzing protein folding. in the ER lumen. And they do that through association of particular intermediates, processing intermediates of the carbohydrate with chaperones, such as calnexin and calreticulin. And then after the protein has attained its proper fold, then the protein is exported from the endoplasmic reticulum into the Golgi apparatus, where then a species-specific array of glycosyl transferases is present that takes that universal mannose-8 GLUTNAC2 precursor and remodels it into species or even sometimes cell-type specific glycosylation structures. So there's a whole array of those, and then the final product would be a glycoprotein, and here represented by the influenza virus hemagglutinin, which is the main antigen in the current influenza vaccines. It's here in complex with a Fab fragment to help it crystallize. But I like this picture because it illustrates that carbohydrates, everything in yellow here is carbohydrate, are really very large modifications that cover up extensive areas of the protein surface. And so if you want to use a protein like this biopharmaceutically as a vaccine, the naive B-cell receptors have almost no chance of recognizing epitopes that are partially shielded by these carbohydrates. And as a matter of fact, quite a few viruses use this as an effective evolutionary strategy to escape neutralizing antibody responses. And the HIV virus is a case in point, but the influenza virus does that as well. So that's why from this biopharmaceutical research line that started, that Walter Fiers started, initially this was all in E. coli, but a lot of proteins will not fold in E. coli precisely because they lack this glycan-associated protein folding catalysis. And hence, in biopharmaceutical industry, we have to use eukaryotic systems to cope with that problem. So intact monoclonal antibodies, for example, which is 60% of the pharma pipeline today, fall in that category. And so we're not using E. coli to make those, we're using mammalian cells. So this is also the context a little bit in which Gobind called upon Roland's expertise, and he knew Roland was working in the group in Ghent on glycosylation engineering, and at a certain point Gobind was trying to express, in fact successfully expressed, rhodopsin recombinantly in HEC293 cells, human embryonic kidney cells. But then a major problem that people were confronted with in using those cells for structural biology was that you get tremendously heterogeneous glycosylation on these proteins. And this is the protein as you would extract it from the rod outer segments from bovine eyes. But the protein you make recombinantly is really tremendously glycosylated. And here is the glycan. It's difficult to see. Really, the way to look at this would be that the sugars would be as big as the protein if they are made in HEC cells. So that was a problem, and then Phil Reeves was working on that, and I just got in touch with him yesterday. He sends his best regards. The reason he couldn't come is that he's really very heavily occupied in teaching, and his university wouldn't let him escape, even for a few days now. So the way Phil and I started working on this is based on a trick. And if you mutagenize mammalian cells, there is a slight chance that you might hit all of the alleles in HEC cells that code for this particular glycosyl transferase called GLUTNAC transferase 1, which is really the gatekeeper in converting high mannose carbohydrates into so-called complex type glycans at the cell surface of mammalian cells. And then ricin toxin has a galactose binding domain, and it has, of course, a toxin domain as well. But ricin toxin cannot get into mammalian cells if there is no terminal galactosides at the cell surface. So if you hit all of the alleles here of GLUTNAC transferase 1, you generate a cell that can survive ricin selection. And so that's exactly the way this was done. And then this results into a protein that is really very homogeneous indeed, pretty much similar to what you would extract from rod outer segments. And then this is a carbohydrate analytical method that I developed during my PhD, and perhaps that was appealing to Gobind because what we used was a sequencing device from Applied Biosystems that had been developed for Sanger sequencing of DNA. So we kind of we labeled these carbohydrates with anionic fluorophores to make them move into the capillary electrophoresis, so in the electric field. And then we used that DNA sequencer with laser-induced fluorescence to map out these carbohydrates linked to the protein. And so what you get here is a Cy standard, which is a hydrolysate of starch. And then this is the glycans that you get from the wild-type HEK cells. As you can see, this is very, very heterogeneous. And then from rod outer segments, you get this profile. And then from this glutamate transferase 1 knockout mammalian cell, we got a very homogeneous profile indeed. And so this generated a paper that I checked yesterday. It has been cited 450 times by now. A lot of structural biologists are using this cell line to cope with similar problems that they face. Okay. So then I want to switch gears a bit, and the rest of the talk will be about yeast. And so yeast has been an organism that we've worked on for the past close to 20 years now to see if we could actually modify the glycosylation pathways in this organism so that they would correspond to what mammalian cells normally make. And the idea here is that at present, the biopharma industry is using mammalian cells, which are really not very robust cells, if you look at them carefully. They don't have a cell wall. They need expensive media to grow. They require extensive control of their extracellular environment to cultivate them. And so if we could manufacture glycoprotein therapeutics in yeast, that may be a reasonable advance and would reduce costs and allow us to do the synthetic biology approaches that we've heard about in a microorganism now, rather than in a mammalian cell, to try and optimize pharmaceutical protein production. And so we've been looking mainly at the types of glycans as you find them on the CH2 domain of human IgG1. So there is a conserved glycan there in glycan of fairly simple structure. So they're biantennary N glycans that carry a core fucose residue, which is important for the interaction with Fc gamma receptors. And then there's partial galactosylation of those branches. So we tried and built yeast to an organism that could synthesize something like this. And so yeasts obviously have a cell wall, which makes them very robust as expression systems, but at the same time causes a bit of a challenge for us because the cell wall is largely made up exactly out of those glycans that we now try to engineer. And so you have to walk a narrow path between modifying them to the human system and yet make sure that the yeast is not suffering from that too much. That's why perhaps it has taken us more than the 2 or 3 years that initially we thought it would take. It's taken us close to 2 decades to solve this to our satisfaction. So the pathway really in the Golgi differs between mammalian cells and yeast cells. What yeasts do is to take this universal mannose-8 glutamate-2 product that comes from the endoplasmic reticulum, and then it extends this with further mannose residues and mannose phosphoryl residues, and really build extensive structures that form part of the cell wall. Now, what we tried to do is to knock out this initial transferase here, and then we introduced a fungal mannosidase to convert to mannose 5. And then we built in the different glycosyl transferases fused, so the catalytic domains from mammalian glycosyl transferases, but fused to the Golgi targeting signals of fungal glycosyl transferases. And in exactly how you make that fusion, and in exactly how you tune the expression levels of those enzymes, that is where the difference is between a proof of concept experiment and something that, in which this pathway is 90 to 95% efficient at every single step. And that took us a long time to accomplish. So in around 2008, 2009, we thought we were more or less done. And here again you see capillary electrophoresis profiles where this is the wild-type glycosylation, MAN5, glutamate MAN5, and then glutamate MAN3, glutamate2 MAN3, and then the galactosylated form. And in fact, we thought this looks like the glycosylation profile that you normally find from CHO cells that make monoclonal antibodies. And Merck Pharmaceuticals bought a company called Glycophy as well that we were competing with at that time. And Merck paid close to $500 million for this at this stage. And we were so surprised because around that time, we discovered that the job was not quite done after doing this. And the point is that if you start looking more carefully at this, what first looks like a clean baseline, there is actually tiny vibrations of that baseline, which indicates still remaining endogenous yeast glycans. And if you add them all up, you end up with 25 to 30% of the total glycan profile. And so what Merck scientists tried to do was to knock out one after the other yeast glycosyl transferase to try and get rid of that background. But in the end, you have a very sickly yeast indeed, and so that didn't get them very far. Even more worrisome, what you sometimes see on particular glycosylation sites, on particular proteins, is that all of a sudden, the human intermediates that the yeast evolutionary has never seen before start becoming substrates for glycosyl transferases that still sit in the Golgi of yeast. Pretty much, I could relate to what Dr. Zhou was saying. These enzymes have never been selected before in evolution to not recognize things that they have never seen before. And so it took us about 2 years to figure out what this was. This is a structure that has never been described before. We're just publishing it. And if we had had the methods of Sabine Flitsch, as you just have heard about, this would have sped up this research tremendously. And so this is very important indeed. So now, after quite a few years, and then, from Gobind, I heard a lot here, and I've been taught by Walter and Roland as well, never ever give up, right? And so one day there will be a new idea that may solve these kind of problems. And indeed, such a new idea came around when we learned that certain fungi actually secrete their glycoproteins, not with this kind of sugars, but with a single GLUTNAC residue on them. And that made us think, oh, they must have an enzyme that can take that off. And that enzyme is expressed in a fungal organism, so clearly it's not toxic to a fungal organism. And so we cloned it, and then with another 3-4 years of work to finally tune the expression of that enzyme, we could manage to express it in Pichia pastoris, which is the yeast that we are using here, metallotrophic yeast. And so in a single step modification, in fact, this is GM-CSF, granulocyte macrophage colony stimulating factor, an important therapeutic. It normally has, it has 2 N-glycosylation sites that are partially occupied. This is the non-glycosylated form, 1 N-glycan and 2 N-glycans. And then you see the heterogeneity typically associated to yeast and carbohydrates. So in the glycodelete, as we call this system now, with a single transformation, we can get rid of all of that. And this is fully compatible with yeast life. It's not always as simple. Sometimes we have tremendously heterogeneous products, like interleukin-22 in this case, which is also proteolyzed partially. But at least we can convert this, like probably thousands of different glycoforms into 3. So this is a proteolyzed form. This is the intact form with 3 times single GLUTNAC. And this is a form which we think is a part of the pro-sequence, the secretary signal that we use there. So at least here you can purify. I mean, you can forget about trying to purify anything out of that there. So we combine that now with what we call this glycoswitch, because it switches from yeast to human. We combine that now, and so it's a kind of like a restriction enzyme, if you wish, for carbohydrates. So everything which remains of the yeast type is now substrate for this endoglycosidase, such that these structures are all converted to a single GLUTNAC. And this enzyme that we use, this glycodelete enzyme, does not recognize the human intermediates. And so those remain on the protein. And so really this allowed us now, as you can see, you see all these tiny vibrations on the baseline and some intermediates there. If we combine this with glycodelete, all of that is gone, and these remaining structures are these intermediates that we synthesize, and are human intermediates. So now, after all this work, I think I now dare say that this system is ready for antibody production in biopharmaceuticals. It was not 10 years ago. So what could we use this for now? And this is a bit of a future perspective, perhaps. I think we've all witnessed the outbreaks of Ebola in Western Africa and the outbreaks of influenza more recently. And there, the drama was that we had antibodies to protect these people. We knew which antibodies to use, but we couldn't roll it out quickly enough because there was no production methodology that would allow us to respond quickly enough to this. Now, the good thing about yeast is that it has a doubling time of 2 hours rather than the 24 hours that you find in CHO cells, allowing us really to make, if we have broadly neutralizing antibodies, which we have, to make these stocks in the freezer. And we're working together with companies, including this one, who are investing in building modular manufacturing plants that sit in shipping containers and in which all the media and buffers that you need are present there in a dried form. The only thing you have to add is utilities, water supply and power supply. And you could churn out the first batches, first thousands of batches in a matter of a couple of weeks. We haven't demonstrated this yet, but the potential is there to do this. And so we're working very hard at the moment, also engineering the antibody format that we produce to make it very compatible with yeast, to hopefully, when next outbreaks come around, to be able to respond much more rapidly than we could today. I have maybe 3 or 4 more minutes to go, and I want to introduce something which I have never talked before in public. And so, you know, after 35 years of recombinant protein production, probably closer to 40 years now, it's still really trial and error if you have a new protein to figure out whether it will express or not. And illustrating that, although we know all of these components in the secretory system, our integrative view of how they work together is clearly not sufficient at this point to generate predictive models of whether a particular sequence will fold in the plasmid reticulum. And so Morgan Bone, who is now just graduated and is with Peter Walter now at UCSF, she has bravely started to tackle this problem by generating a new method. Not to study secretability of 1 or 2 model proteins, but to try and generate secretability data across the proteome. And so she's worked for 5 years to design a new type of cDNA library where we are generating domain-sized protein fragments across the proteome. And we're focusing on single domain-sized fragments because if you have a big multi-domain protein, the presence of a single domain that doesn't fold will kill the secretability of that protein. So we chop it up in domains. And what we then use is yeast surface display as a proxy for productive passage of a protein fragment through the eukaryotic secretory system. So in yeast surface display, you fuse the protein at its C-terminus to an anchor protein that by nature is incorporated in the outermost layers of the yeast cell wall. And so the protein entirely transverses the secretory system. And because the fusion is at the C-terminus, the impact of the C-terminal fusion partner on the N-terminal, on the folding of the N-terminal part is minimized, is relatively minimal. So the trick was here to really be able to capture the entire human proteome into a yeast surface display library that had just a few million to a few tens of million clones. And so that is technical, and I will skip it, although it's very important. So she designed an entirely new cDNA preparation method, which incorporates normalization of the cDNA library in combination with random priming and starting from fragmented messenger RNA. And so this has now generated a library that overlaps the size range of human domains. We would like to get this a little bit bigger still. So the median size of an independently folding human protein domain is in the neighborhood of 85 amino acids. And so that's what we're trying to cover. So then we sort these libraries. We induce expression, and we sort these libraries using a combination of magnetic activated cell sorting and fluorescence activated cell sorting for those protein fragments that show evidence of being secretable. And so from about 2 million clones, so our proof of concept libraries are about 2 million clones, we can get expressibility data. So we do this in triplicate, and we only consider something secretable if we see it secretable in the 3 different replicates that we have. So from 2 million, we can get expressibility data of about 150,000 protein fragments, hitting about 25% of the entire proteome at least once. And so we now have to extend this, amplify it maybe 25-fold or so, to really get to very decent levels of coverage of the human proteome. So what this allows you to do is to generate catalogs, databases, where any of you could go and have a look. If you're going to express a particular part of a protein, and you want to make an antibody, for example, that recognizes a conformational epitope, you could go into the database and get experimental evidence on whether you can actually make that. So Other applications are, of course, more fundamental in nature, where we've seen now that a substantial fraction of what we see has a PDB hit, and that allows us then to do data mining on a structural level to see which features of proteins are enriched in those that can be secreted versus those that cannot be secreted. And sometimes you come up with very peculiar cases, like these are two different members of the same protein family. This one is non-secretable and that one is secretable. And that now, through data analysis, data mining, hopefully will allow us to extract rules and predictive models of what we can make. Perhaps that's naive. We will see in a few years how far that will go. So we're collaborating with groups that are in deep learning. These are the new methods that are used in facial recognition in self-driving cars, right, to do feature extraction, automated feature extraction, non-biased by any insight of the human mind, unfortunately, to see if we can see patterns there and predictive value. So that is my group, a very, very good group at the moment, working on biopharmaceuticals and bioanalytics, but always starting from basic research and building our own tools. I think that's something that many of us here in this audience share from Gobin's legacy in the beginning. So this is Gobin's at a retirement ceremony we had for Walter Fierce in 1997, exactly 20 years ago now. And what I recall, I was a grad student at that moment. The secretaries who were organizing this meeting, they told me, you know, the person, there were a few prima donnas that came, you know, who were very hard to, you know, we had a lot of work with them. But Gobind, you know, he came in economy class, you know, so his ticket was the cheapest that anybody of the people who came. And we had not a single problem with him at all, you know. It was just, that reminded me, and when I then met him finally in 2000, I could clearly relate to that and understand what they were talking about. So thank you very much. That was all I had to say.
Thank you for this inspiring talk. Questions? No question? You don't want to really investigate any of his libraries? Oh, yeah.
With this new delete strain, you would of course put some human-like glycosylation on proteins, and that would make the proteins more stable, presumably by making them less amenable to proteolytic degradation.
Yeah.
But would you necessarily replicate the natural glycosylation that's necessary for folding to occur?
Yes, so that's exactly what we do.
And also the antigenicity that might be involved with some of the glycosylation if it is non-human, if the enzymes end up putting things a little different.
Well, apparently I didn't make myself clear all too well. So what we did is exactly mimic the glycans as they are found on the human IgG that is in all of our blood. So the sugars that we have on there are exactly the same ones, except for this proportion of single glucnac that we generate now by doing the glycodelete engineering. Yeah. But that, in fact, if you look in the proteomics data sets that are there today in the databases, you do find glycopeptides that have single glucnac, and they originate in lysosomal degradation of glycoproteins. And so this is not as unnatural as you might think in the first place. And so the glycans are not only important for stability, they're also very important on antibodies for triggering Fc gamma receptor dependent effector functions. And so yeast does not make core fucose on those glycans in the first place, so you don't have to engineer it out. And if you have non-core fucosylated glycans on an IgG, the antibody-dependent cellular cytotoxicity effect goes up by a factor of 100. And so if you have virally infected cells that need to be killed off, or tumor cells that need to be killed off, that's exactly the glycosylation profile you want.
Any more questions? Yes.
Why do you choose Pichia instead of the Saccharomyces?
Right. Well, so Pichia pastori is a metallotrophic yeast, and it's there's a few reasons to use it. First of all, its N glycans by nature are much shorter than those of Saccharomyces. So if you start engineering it, the impact on the physiology is smaller than if you do that in Saccharomyces. So that's one reason. And then the second reason is that it's a Crabtree negative organism, so it doesn't ferment. Saccharomyces generates ethanol and kills itself at a certain point, right? And so with Pichia, we go to 400 grams of wet cell weight per liter here. So it's very high cell density. All right, thank you.
Thank you very much. We will have one last speaker in this session. I hope I can pronounce it correctly. Kivyon Cha, who is acting president of a company that he's going to explain. And
it's a public organization.
A public organization, okay. And the title is Coordinated Development for New Global Innovative Biotherapeutics in Korea. Please.
Okay, thank you. And thank you for inviting me to this wonderful conference. Actually, I was the last person who enrolled this conference, and then I also thank you, Tom and Razinda, to squeeze in my session into this lovely conference. And also special thanks to two Gobins facing each other. Okay. Okay, to make the long story short, I brought very short video clips, which I'm working on. My institute is a Korea government-funded public organization. So what we are doing is that you have done a lot of the mechanism-based research. So we call it mode of actions. What is going to be beyond that? So actually, Nico and so many people has been introducing what's going to be the biotherapeutics after you are studying a lot of mechanistic work. So 20% of 30%, when you go to the FDA to get the approval, 20% of and 30% is very important. Your mechanistic work is very important, okay? I cannot say it is necessary, but it is very crucial. Without it, probably 10 times harder time you will get it. But the leftover 70% to 80% is not something to do with the mechanistic. It is going to be safety issues, and then the glycosylation, and then there's processing issues. Okay, my institute is to solve that black box after your research, before the company is getting in. Okay. Oops, sorry. Okay. Can you make a voice? Okay.
Where does the strength that raises national competitiveness originate from? What do companies that dream of success need the most? Our inquiries meet the world like this. The place becomes the bridge to success for a medical industry and an economic growth in Korea. The new history begins in Osong Medical Innovation Foundation. The best way to predict the future is to invent it. Medical industry, the industry that the world pays attention as the next generation technology following automobile, IT, and shipbuilding. Noticing the value and infinite possibility of advanced medicine since the early 90s, Korea has started creating Korea's first medical complex, Osong Korea Medical Cluster, since 2009 as a part of National Project. We didn't pause at the first step. Settling the world's only R&D cluster where companies, universities, research institutes, and biohealth area, national institutions are concentrated. Osong Korea Medical Cluster has proved its potential. Osong Medical Innovation Foundation stands as the center of incalculable growth. Osong Korea Medical Cluster has experienced a rapid growth with an unstinting R&D support by Osong Medical Innovation Foundation. It is not a coincidence that global companies chose Osong Medical Innovation Foundation. Tomorrow belongs to those who have vision today. Osong Medical Innovation Foundation provides one-stop service for R&D. New Drug Development Center, which develops and optimizes new drug candidates to promote the development of new biomedical drugs. Medical Device Development Center, which supports all stages from the R&D of medical devices, production of test product, to trials in one stop. Laboratory Animal Center, which provides world-class system for preclinical evaluation of newly developed drugs and medical devices. And Clinical Drug Manufacturing Center, which manufactures biopharmaceutical products according to GMP. The 4 core research support facilities support the entire process from product development to commercialization. Also, through the Clinical Research Hospital that will enable clinical trial, it will become the world's only biohealth cluster that will support from the basic R&D support to commercialization in one stop. Located at the center of the nation, Osong Korea Medical Cluster will reduce your cost and time through outstanding transportation network based on its geographical conditions. Easily provide necessary information for R&D through national research support facilities. And 6 public health administrative organizations near the cluster offers more convenient licensing processes regarding products, including R&D and manpower training. Differentiated incentive reduced companies' financial burden and eliminated inconvenience in institutional support. The place that completes global-class infrastructure in medical industry from R&C support to licensing and commercialization. Osong Medical Innovation Foundation will complete Osong Korea Medical Cluster as the new role model of medical industrialization and lead companies' development.
오송 첨단의료산업진흥재단은 의료산업이 대한민국의 차세대 경제성장 동력이 될 수 있도록 글로벌 수준의 바이오 신약과 첨단 의료기기 연구를 개발을 지원하는 기관입니다. 저희 재단을 통해서 첨단 의료산업이 대한민국의 국부를 창출하고 대한민국의 국격을 더 높일 수 있도록 하겠습니다.
I'd rather attempt to do something great and fail than to attempt to do nothing and succeed. And now we face the future again. We challenge towards the new tomorrow without rushing or giving up. At the place where state-of-the-art companies in Korea and worldwide chose and Korean government supports. As the global advanced base of medical industry that leads Korea's economic growth and job creation. To become a global biomedical hub that leads the world. Wait and see. The place where your success, Korea's growth, and global medical industry's success happen. This is Osong Medical Innovation Foundation. Osong Medical Innovation Foundation.
Knowledge city and then scientific city, and you are also planning for. And then 10 years ago, Korea has started to do this biocluster projects. Okay, $8 billion supposed to be invested to establish, and then right now the $1 billion has been already put it in. So not only all the building, now after groundbreaking, actually it's been 4 years to develop all the technologies to do it. So I'm not gonna overlap with the video clips. What I can do is something short talk without any overlaps. This is a personal picture. This is a personal picture with the Govind and Esther over here. And yesterday I asked my wife. Okay, many people is showing their pictures. Let me please send me. Okay, some the picture with Govind, and then this is my wife's collection. It's not my scientific collection. So Govind and Esther is there, and then when I come to Boston, 1994, actually after 1 year walking, we had my baby's first year birthday ceremony, and then I asked Govind then to invite, and then Govind and Esther was showing together. Then with my dad and the mom with my wife, actually is there. So this is a lovely picture. So what also Govind is like a family father for me. Okay, this is again the this is my wife's collection. So the always with the kids. So actually, when my professor, I have two great scientists, and Govind and my previous supervisor, PhD supervisor, Professor Im. Actually, he visited with his daughter to the Boston, and then Govind invited us to the Rockport, and then had taken a picture. Actually, Esther has taken these pictures. So I also thanks to Esther as well as Govind. Okay, what we are doing is to support and promoting your mechanistic work, your basic science work, to go to the end of the clinical therapeutic development. Okay, this is all the overlaps. Korea has designated the ozone as bioclusters, like Chandigarh did for this knowledge city. So the FDA and then KCDC and the 6 government administration, the offices has been moved from the Seoul to Ozone, and then they made a core infrastructure which I'm working on, K-Biohealth. The idea is that this is overlaps, new development, new drug development center, medical device development center, and then laboratory animal center, and then the clinical drug manufacturing center, which has a biotherapeutics at the GMP level. Then we call it one-stop service. When professor coming or company people coming, no matter what the size of a company, if you have a target, if you have a protein biotherapeutic, the hits, now we can optimize it before that can be injected into the human body. So Govind used to say, right, Kiwon, you have to make sure, you have to be very sure for your compounds, right? Same things goes on. We have to be very sure for biotherapeutics before we inject into the human body. Everything has to be done. Everything has to be resolved before we get the side effect from the patient. So that's why this one requires $1 billion per one product. 3 years ago, now they say $2.6 billion per project. That requires the biotherapeutic development. But once you are successful to do that, now you are gonna have 50 or the $70 billion can be obtained. Here is the problem. Okay, this is what you are doing in the academia and institute of R&D pipeline. You have your own ways, mechanistic ways, doses, and then this is what company is doing. Sometimes companies started from the R&D, and then this is for the commercialized drugs. There's huge gaps what academia people is thinking and the other company developing people is thinking. So usually big pharma like Pfizer, Merck, and Sanofi or Novartis, they have their own specialists and their own apparatus and then technology to fill it up. No other company has it. And then unfortunately Korea, I don't think India, they don't have apparatus or technology to fill it in. So Korea government, actually 3 ministries comes together to solve this problem and then put some investment to solve it, to help the resources and then the professors and then company. This is the area what we are focusing on. Target validations and then mechanistic studies. Professor is doing so good job. I think all of you are doing a very nice job in your life. And then company is taking care of the last process of this drug development. There is a huge black box here. So only big pharma is doing globally, but Korea make a decision to. When you have targets, hit compound can be made by phage screening or protein engineering, okay? And then there is optimization process. No single product can be injected without optimization into human body. So very thorough process need to be done before that can be injected. That called it the lead compound identification and the optimization and candidate. Means, okay, I will do that. So this is something public area need to be done. The strategic plans and all the process and then technology can be explained whenever all of you guys can visit Korea or visit our institute. Many things. How the FC engineering, protein engineering for optimization. And then all the validation-based efficacy evaluations, okay? Mass spectrometry-based evaluation, and then more or less a safety evaluation for the animals and cells, and then the immunogenicity, what we established for the glycosylation, those things, okay? And then process development to get the grams of protein out of liter. So with the Samsung manufacturing, they have a 360,000 liter reactor with a productivity of more than gram per liter. Okay, so the other one I can explain whenever you have a time to visit us, Korea, and our institute. Okay, thank you. So everybody is hungry. Okay, thank you.
Thank you very much. I have one question.
Okay.
Who owns the final intellectual property?
Okay, very important question. So, right now we are giving the company. Basically, the basic idea is we are not making a problem before they have a final products, whatever intellectual properties or the other one. So far we are giving if they have their targets, we are giving the services, no intellectual property. But some small company like to share the profits, then we have reduced the prices to the debt.
If you have your professor in this red image that you have there,
okay,
and his or her graduate student,
I'm sorry?
His or her graduate student, can they publish their result?
Oh, yes, you can, but this special area, professors sometimes give up the publication. But there is some field we need to develop the new processes or the protein engineering field, then we can publish. But But it's not like a mechanism based. So that's possible.
I'm really very impressed. But also we have to develop new processes for intellectual property publication.
Right.
I thank everybody who spoke this morning, and we are now going to having a picture taken, or where do we go?
Yes.
Thank you.
Okay, thank you.
Thank you, everyone. Now we'll start the next session at 2:50. Now we'll have a group photograph that will be in the entrance of this building downstairs. Thank you.
सर आपके पास हमारे पास है ना ये फ़ोटो सेशन है अभी